Hey guys, so today we're starting our fourth and final unit. This unit is all about the human body, so we'll be going over a bunch of different systems in the human body, starting with the digestive system. Today we're going to be going over nutrients. So for this lesson on nutrients, we're going to be going over the different types of nutrients. You need to know the structure of different types of carbs, as well as different types of lipids and proteins. You need to know how all of these different types of nutrients are synthesized through a process called dehydration synthesis as well. So the different categories of nutrients are carbohydrates, or carbs for short, lipids, proteins, vitamins, and minerals. Nutrients are really just chemicals that are required for life and have to be absorbed and assimilated into the body of all living things, plants and animals. So the first is carbs. Those are made of sugar molecules that contain carbon, oxygen, and hydrogen. Some examples of those are like glucose and sucrose. They are the primary source of energy for cells. The energy in carbs can be really readily available because carbs can be turned directly into ATP energy through cellular respiration. They do also make up the largest component of most diets. They include things like potatoes, bread, corn, rice, and fruit. If you have too many carbs in your diet, those excess carbs that are not being used up as energy will be stored as fat. So that's why some people might cut carbs out of their diet if they're trying to lose fat. The human body cannot make carbs. It has to be ingested, and carbs are made via photosynthesis. And then we as humans can only get them by eating plants. Carbs can be either single units or polymers made up of numerous sugar units all connected to one another. Each individual sugar unit will contain a carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So what that means is that there will be usually twice as many hydrogen as there is carbon and oxygen. So for example, glucose is C6H12O6, so equal amounts of carbon and oxygen and twice as many hydrogen. Carbohydrate names tend to end with OSE, so like glucose, sucrose, fructose, cellulose are all examples of carbohydrates. And we classify carbs based on the number of sugar units that they are made of. So the first type is monosaccharides, and they only have a single sugar unit. So they are the simplest types of sugars. They include glucose and fructose. So this picture here, this ring is a single glucose unit. This is a single fructose unit. The images above them are just what it would look like if you like unlatched the circle and stretched it out into a straight line. These sugars are isomers of one another, meaning that they have the same chemical formula, but different structures. So you can see by looking at them that they do look different, but they both have 6 carbons, 12 hydrogens, and 6 oxygens. The properties of isomers are going to be different from one another. The structure does matter, not just the chemical formula. So for example, fructose is sweeter than glucose. The second type of carb is a disaccharide that will have 2 sugar units or 2 monosaccharides attached to one another. This includes sucrose, which is made of a glucose attached to a fructose, as well as maltose, which is 2 different glucose units stuck to one another. The way we create disaccharides or any type of polysaccharide is a process called dehydration synthesis. Essentially, you remove a hydrogen from one of your monosaccharides, a hydroxide from the other one. That hydrogen and hydroxide, which is OH, combine to form a water. So we remove the water from the molecule. That's why it's called dehydration, because we're removing water. And where that water is removed, a bond will form. The opposite process of this is hydrolysis, hydro meaning water, lysis meaning to break apart. And this is when a water molecule is added to a bond in order to break the bond apart. That is how we would go from a disaccharide to monosaccharides. So this is what it looks like. Essentially, you have 2 different monosaccharides here, so 1 and 2. I'm going to take a hydrogen from one of the monosaccharides, a hydroxide, which is an OH from the other one. Those combine to form a water, and that water is removed. That's the dehydration part. And wherever that H and OH were removed, a bond is going to form there. So this oxygen here is now going to form a bond right here where the OH was. That is what this bond that we see right here is. It's where that water was removed. The water is then just a byproduct. Yeah. Okay, polysaccharides are types of carbs that just have more than 2 sugar units bonded together. So for example, starch is a polysaccharide that has thousands of glucose units all attached to one another, and they can be either branched or unbranched. Animals tend to store carbs in the form of the polysaccharide glycogen, which has 16 to 24 glucose units. Plants tend to use both things like starch in things like potatoes, as well as plant cell walls are made up of something called cellulose. So this is just the difference between branched and unbranched. So an unbranched polysaccharide will just essentially be a straight line, whereas a branched polysaccharide will have like different lines that are attaching to one another. Okay, so that is it for our carbs. Next up is our lipids, which are also called fats. Lipids are non-polar, which means that they are insoluble in water, so they don't dissolve very well in water. They are made up of a glycerol molecule as well as fatty acids. The glycerol is bonded to the fatty acids via dehydration synthesis, so the same process that we use to make polysaccharides and disaccharides. Again, we're going to take an H away from one part of the molecule and OH away from another part. That forms our water that we remove, and a bond is going to form there. So for example, we have triglycerides, where essentially you take a glycerol molecule and you bind it to 3 fatty acid molecules using dehydration synthesis. You remove 3 waters essentially. Lipids store energy, so they can be used as an energy source in cells, but the energy is not as readily available as carb energy. However, like we talked about when we were talking about carbs, if you have too many carbs in your diet, those excess carbs will be converted into fat to become stored energy. Lipids are also used to make hormones. They carry fat-soluble vitamins, and they make up your cell membrane, so they're important for a lot of different body functions. They're also important on the human body for things like insulation. So one of the types of lipids is a triglyceride. Again, like we said, it is a glycerol bonded to 3 fatty acids. Triglycerides can be either saturated or unsaturated. A saturated triglyceride, also called a saturated fat, is essentially when you only have single bonds between the carbon atoms of your triglyceride. If your triglyceride is unsaturated, so it's an unsaturated fat, then it would contain double bonds somewhere in the molecule. It doesn't have to have all double bonds. If there's even just a single double bond between the carbons of the triglyceride, it is then unsaturated. Unsaturated because if we broke that double bond apart, each of the carbons would be able to bond another hydrogen. So a saturated triglyceride is full of hydrogens. There's nowhere on that molecule you could bond another hydrogen. With an unsaturated triglyceride, where it has some of those double bonds, you could just break open that double bond and you'd have 2 more bonding sites from hydrogen. So it's not saturated with hydrogen. Saturated fats tend to be more stable, so they are often solid at room temperature. Unsaturated fats are less stable, so you usually find them in liquid form at room temperature. And because of that, we sometimes just refer to them as oils. So this is what a triglyceride would look like. This would be kind of its final form here. This purple area is the glycerol, and then each of the red areas are our fatty acids. On the left-hand side here, it's showing how this is made. So we are going to take 3 OHs away from our glycerol, 3 H's away from our fatty acids, and then each of those becomes a water molecule. Anywhere that we then remove that water molecule becomes a bond, which we can see as these 3 bonds over here. And this is just an example at the bottom here of the difference between a saturated and unsaturated triglyceride. If we look and our carbons are attached all to one another with single bonds, then that is a saturated fatty acid. If between our carbons we have areas where there are double bonds, now it is unsaturated. And that's because this carbon here, for instance, and this carbon here could hold more hydrogens if we just broke that double bond. The other main type of lipid is a phospholipid, which you might remember from Science 10. Phospholipids are what make up the phospholipid bilayer that makes up all cell walls, sorry, not cell walls, cell membranes. And this is slightly different than the triglyceride because in this case, the glycerol is only bonded to 2 fatty acids instead of 3, but it's also bonded to a phosphate group, hence the phospholipid. So the glycerol is still bonded to 3 things, but instead of all 3 of those things being fatty acids, only 2 of them are fatty acids, and then the third is a glycerol. The phosphate is the head, and it is polar. That's what makes it hydrophilic, meaning it likes water. The 2 tails, if you remember, are hydrophobic. Those are the fatty acid chains. They will be insoluble in water. And again, this is just kind of a picture of what it looks like. So right here, we would have our glycerol. The light circled part up here is the phosphate, and then we have the 2 fatty acid tails sticking off. This is just another image of what our phospholipid would look like. Whoopsies. Yeah. Wax is also a type of lipid, though we don't talk about it as much because we don't really eat a lot of waxes. Waxes are made up of long chains of fatty acids bonded to either carbon rings, I should say, not rinds, rings, or alcohols. They're very long. They're also very stable. They tend to be insoluble. So that's why, like you might put wax on the surface of something that you want to be waterproof. It's also why you have like the waxy cuticle on tops of like lots of plant leaves so that water doesn't evaporate out of them as easily. That kind of stuff. So those are examples at the bottom. Again, I don't really care about waxes as much as the other 2 types of lipids. Then we get into proteins. Proteins are chains of amino acids that form structures in cells, and there's millions of different structures in your body that are made up of proteins. Pretty much all of your organelles, antibodies, enzymes, like muscle tissue, all that kind of stuff is made up of protein. Your hair, your skin cells, all of that. Antibodies are just specialized proteins that are part of the immune system. We'll talk about that when we get to the immune system portion of this unit. Enzymes are special proteins that speed up reactions. We'll talk about them in the next lesson. Proteins are usually not used as a source of energy. However, if you completely run out of first your carbs, your short-term energy supply, and then also burn through all of your stored fat reserves, which would only happen if you were like literally starving for weeks on end, then you might get to the point where you start breaking down some of your protein and start breaking down your muscles essentially as a source of energy. Amino acids are the building blocks of proteins. So amino acids are just a molecule that is made up of an amino group bonded to a carbon, bonded to a carboxyl group, which is COOH, and then bonded to some other group, which is called the R group. There are 20 different possible amino acids in the human body. Proteins are just made by linking together these amino acids in different combinations. Some proteins are fairly simple and only contain a few amino acids, and others can be hundreds of thousands of amino acids long. The order of the amino acids, the types of the amino acids, and the number of the amino acids all determine the type of protein that is going to be formed. And those proteins then determine your traits. I mean, your like hair color, for instance, is based on the proteins of your hair. The human body can make some of its own amino acids, but there are 8 amino acids that your body cannot make. You can only get them from eating things. Those are called the essential amino acids because it's essential that you have them in your diet. So this is what an amino acid looks like. So you have this central carbon here. It is bonded to a hydrogen, to your amino group, to your carboxyl group, and then to the R group. And the R group is the part that like changes all of the time between the 20 different amino acids. You do not need to know the different amino acids. Protein synthesis is really important. It's how we make a lot of our different cells and organelles and stuff like that. So what happens is you eat and digest the protein. When you digest the protein, it breaks apart into all the individual amino acids. Those individual amino acids are absorbed into the bloodstream, carried to your cells, and then at the ribosomes of your cells, which are the organelles responsible for protein synthesis, all of the amino acids will be assembled into different proteins based on the proteins that your DNA codes for. Amino acids are joined together via the process dehydration synthesis. So the same way that we make our fats and our carbs is also how we make our proteins. So we take an H away from one of the amino acids, an OH away from the other amino acid. That becomes our water that we remove, and that is where the bond is formed. This bond with carbs, lipids, and proteins is always a covalent bond. So with proteins, we have a special name for it. We call it a peptide bond, just because a chain of amino acids is called a polypeptide. So the bond in the middle of it is called a peptide bond. The polypeptide is then just like folded up to make the final product of the protein. So once again, this is what it would look like. So we have 2 amino acids here. We take an O away from one, which could be an OH there, and then an H. That's going to be our water that we remove. A bond formed wherever we have done that. Now you'll see this one looks a little bit different. There's a minus sign here instead of an H, and there's an H plus here. That's just because this H plus should be over here, but if this is happening in water, it moves. You don't really have to worry about that as long as you know a water is being removed during protein synthesis. Okay, so like I said, once the chain of amino acids, which is the polypeptide, is formed, it needs to be folded up to become the protein. And so there's 4 different levels of protein structure. The primary structure of a protein is just the sequence of amino acids. So what order are the amino acids in? The secondary structure is any hydrogen bonds that form between the amino acids that aren't beside each other. And when you have those hydrogen bonds forming, you create either something called an alpha helix or a beta pleated sheet. A beta pleated sheet is like if you've ever like taken a piece of paper and like accordioned in it, where you keep accordioned it, where you like fold it back and forth on itself. That's what a beta pleated sheet looks like. An alpha helix is just like a spiral. Then the tertiary structure is when you essentially fold it up even more and form like covalent bonds between alpha helixes and beta pleated sheets. And then the quaternary structure only exists for some proteins. Essentially, if you take 2 different polypeptides and attach them to make like a super protein, that is your quaternary structure. So this is kind of what that would look like. So just the order of all of our amino acids here is the primary structure. Then we create some hydrogen bonds to create either a beta pleated sheet or an alpha helix. Then you'll have covalent bonds forming between beta pleated sheets and alpha helixes, which further folds up your protein onto itself. And then if you have kind of 2 folded proteins attached to one another, that would be a quaternary structure. Proteins can both denature and coagulate. Denature is when a protein temporarily changes shape. Coagulation is when a protein permanently changes shape, and both of these can cause the protein to not function. When the protein is not in its correct shape, it doesn't work properly. This usually happens when the bonds within the protein, like all those hydrogen bonds and stuff, are disrupted, which can happen if the proteins get too hot. So this is why like really high fevers can be dangerous. It can also happen if you're exposed to like radiation, so you get like radiation poisoning or radiation sickness, or like changes in pH in your blood, which is why it's important that our body maintains homeostasis to maintain things like your blood pH. Okay, the last two types of nutrients are your vitamins and minerals. They're needed in much smaller amounts than carbs, fats, and proteins, but they do both help chemical reactions take place in your cells. Vitamins can be either fat soluble, which means they dissolve and are stored in your fat. So that includes vitamins A, D, E, and K. Fat soluble vitamins you can technically overdose on, though it would take a lot of most of them. Water soluble vitamins are things like vitamin B and vitamin C. These ones just dissolve in water. So if you eat or like take too many of these, so for example, like too much vitamin C tablets or something, if you're afraid you're getting sick, the worst that would happen is you might have like loose bowel movements. But yeah, you'll probably just pee it out. Vitamin B, for instance, if you take too much of that, it's going to change the color of your pee, but it is just getting peed out. And then minerals include things like calcium, magnesium, phosphorus, sodium, potassium, iron. Some of these are important for like building your bones, like calcium. Other ones like sodium and potassium are important for things like neurons, where you have to like change the electrochemical gradient of a neuron. Iron is really important for your blood. Okay, that is it for today's lesson. Please let me know if you have any questions.